Town, and I have the pleasure of introducing our main speaker for today, Christian Hellriegel. So Christian studied chemistry and biochemistry at the Ludwig Maximilians University in Munich. His highly interdisciplinary doctoral thesis on single molecule spectroscopy and dynamics in host guest materials was conducted at the Center for Nanoscience Excellence Cluster of LMU Munich under the supervision of Prof. Dr. Christoph Brauch. These studies amalgamated biophysics, ultrafast femtosecond scale dynamics, single molecule spectroscopy and drug delivery systems, and host guest materials. As a result of highly fruitful collaborations with the CNRS in France, the Max Planck Society in Germany, he was able to publish over 12 articles in this time, and for this he was awarded the Roma Prize for Best Doctoral Dissertation in 2005. During his postdoc at the Laboratory for Fluorescence Dynamics with Prof. Dr. Enrico Graton at the University of California, Irvine, he employed single molecule techniques primarily to achieve real-time 3D single particle tracking, multi-parameter spectroscopy, and there he endeavored into the realm of fluctuation analysis and multi-photon microscopy design. He left UCI to start a microscopy facility project at the Cardiovascular Research Center, the CNIC, in Madrid, Spain, and there he applied advanced molecular imaging and fluctuation techniques in intravital studies in cardiovascular research. Chris then joined Carl Zeiss Microscopy in a headquarter-based function as a specialist consultant in the global support team, the global sector marketing and life science segment responsible for all advanced microscopy modalities, including super resolution and light seed-based techniques. Since 2018, he is the embedded microscopy specialist at Harvard Center for Biological Imaging, the HCB. In his spare time, Chris thinks he is an accomplished ukulele player. That's the product of lockdown, I'm sure. So Chris, it's great to have you with us. You're clearly a specialist in many things. And today, I believe you're going to speak to us specifically about super resolution microscopy techniques. So over to you. You can start sharing your screen.
Okay, let me start sharing my screen. And first of all, extend my thanks to the organizing committee. And it's always a little bit daunting to hear these introductions because it's quite lengthy. I think I should submit shorter bio sketches. It's just a product of length of time. I've been around for quite a while and did a lot of stuff. And I think, yeah, maybe it would be okay to shorten the list a little bit. It sounds way more impressive than it actually is. All right.
It means that we know all your expertise now, so it's great.
All of it, including ukulele. Okay, can everybody see my screen?
Yes.
Yeah.
Okay, then, well, I'm honored to be invited to speak to this audience about super resolution techniques, and especially so since the most recent advances in optical microscopy, as I will show in this image sequence here. As it starts, well, did it start? Okay. Now allow scientists to access the frontiers of resolution, the frontiers of what can become observable, also dynamically, that is in living organisms. Time constraints will not allow us to go deep into the details and also not explore the whole width and breadth of super resolution microscopy techniques. So we will stay focused on the most recent and promising developments. For completeness, I will be providing references and links to relevant publications that can be accessed to go deeper and broader into the topic of super resolution. If you allow me to play back this video again, what we see here in the magenta color is the stain for the endoplasmatic reticulum, and what we see here in green is the is an outer membrane protein of mitochondria. And what we see here is that intricate orchestration between the mitochondrial movement and it passing through. Look at this example here, it trying to poke through this endoplasmatic reticulum. But anyway, I think what this illustrates is that, you know, with new observation capabilities, you start to obtain new questions that you might be interested in at least looking at to generate nice images for a presentation, or actually examining and figuring out what the hell is going on in there. In this talk, I organized it roughly into an introduction where we're going to look at definitions of what is resolution and what is the so-called super resolution. We're going to take a look at an overview of the methods in a side-by-side kind of comparison. And then I will lead us through the three main families, say, of super resolution techniques with a couple of examples, and then finish off with a brief summary. There are probably several different ways to step into the realm of the motivation behind microscopy, but here I chose one where resolution plays a particularly important role. And this is the work of Ramon y Cajal at around 1885. Here in these pictures, we can see him. His pictures, his micrographs are famous also because of the artistic nature of it. He's you know, on top of being a brilliant scientist, he was also probably an accomplished artist. And we see him here also posing next to his microscopes. His drawings, they came from a time, we have to remember, where there were no digital cameras. Micrographs were difficult to come by, and aside from learning all science and neurology, an accomplished person in that time had to be very resourceful, able to accurately stain tissues, and also be a masterful artist in order to, you know, the super resolution was dictated in those days by the resolution that you would obtain with your own eyes and the ability to describe what you were seeing through an optical microscope. And the astonishing part about Ramon y Cajal's work is that, you know, a lot of his conclusions were drawn from the structural aspects of the neurons he stained, and that they remain very, very accurate and relevant until this day. Okay, we move forward almost a century, and we add another dimension to this entire problem of resolution, which is the dynamic, is that we want to see, you know, dynamic phenomena. And we also remember that a lot of what we see today in children's books of dynamic phenomena are relatively recent pieces of knowledge. Here seen, for example, on this video sequence, that's the spermatogenesis in cricket. So this is a meiosis step where using the then new phase contrast technique in microscopy and ordinary film to capture the cell division. And with the advent of fluorescence and fluorescent staining techniques and the whole biology going behind fluorescent proteins and things of that nature, we opened up a new avenue of microscopy and interrogations in the reign of microscopy, which is the capability of staining cells specifically. And we see here, again, the cell division phenomenon with cells stained with different colors from about 1995. So microscopy has come a long way. And we have here, you know, resolution in space with the addition of the generation of biological and biochemical contrast, that is the fluorescence in this case. That is what propelled microscopy to the limits of what's observable in space and time. And now let's take a look at what does it mean, the limits of space, which is what super resolution is all going to be about. And to that end, I have prepared a slide here. Resolution is in itself a relatively blurry concept. If you look up in the dictionary, the definition of resolution is the analysis into clear-cut components, the capability to distinguish between two or more objects. In microscopy, the distinction is done in space, but we could also consider time, color, polarization, and other aspects. What we see here are three images side by side of a neuron in brain tissue stained with a fluorescence membrane marker. The difference between these three images is only in the acquisition method used to generate the final image. To the left, we see a conventional optical micrograph, as we would see through a conventional wide field microscope with a very good objective. In the middle, there is the resolution enhanced to the level of what is achieved by modern laser scanning microscopes, the confocals, which could be understood as the image at the resolution limit. So if you take the classical resolution limit, the image in the middle is where you would have that classical limit. To the right, we see an image of the same object acquired with a super resolution technique that I will introduce a little bit later called lattice SIM, structured illumination microscopy. So seen from a zoomed out perspective, we almost don't see what is the big advantage of super resolution microscopy. The three objects look quite similar. It becomes apparent when we zoom in. To the left, again, the wide field image with its blurriness, we ascertain that we are looking at individual vesicles there protruding out of that particular cell. In the middle, at the limit of what is possible, we can start to identify individual details in the staining of this membrane. And to the right, we can appreciate even more detail. And as we zoom in more and more, we can see more and more detail. And in the middle here, I added the scale bar of 250 nanometers, which is the resolution, well, about the resolution limit. If we talk about the resolution limit, we invariably would collide with the works of Ernst Abbe and his famous equation, where we see it's carved in stone in a monument somewhere in Jena. And the formula is thus d, the distance, the smallest distance between two objects so that they become resolvable, is the wavelength of light divided by twice the numerical aperture of the imaging system. And for high NA objectives, this limit in practice is found to be around 250 nanometers. So if you're using 500 nanometer light, half of that is going to be 250 nanometers. And this is widely regarded as the resolution limit. For practical reasons, and because none of the optics detections, etc., are ideal, this limit is already pretty hard to achieve. Usually you will have microscopes, or ordinary microscopes will have resolutions that are slightly worse than that. And very, very good microscopes achieve that very limit. So any technique that goes beyond that limit is there's some discussion in the field what is real super resolution and what is not. But the wide consensus is that techniques that go beyond 250 nanometers, say like in the hundreds or in the few dozens of nanometers, those are termed collectively super resolution techniques. Okay, then let's take an overview of what are these super resolution methods, which are the super resolution methods that exist. And I would like to draw your attention to two important publications. One seen here on the left can be downloaded at Wiley. It's those essential knowledge briefings, where the key idea is to introduce an audience, a beginner style audience into a specific field. They have many different types of essential knowledge briefings, and there's one devoted to super resolution microscopy. It contains literature, it's the entrance of a rabbit hole, if you will, where you will get a hand-waving kind of explanation of what super resolution microscopy methods are, and take you deeper and point you to where you could find more information. Another interesting and a little bit more thorough paper is here seen on the right side. This is Lothar Schemelle, Markus Sauer, Chris Eggerling, and others in Nature Cell Biology from a couple of years ago. And here the idea, and it's called super resolution microscopy demystified, because there is a lot of uncertainty, especially from the people that are not, full-blood microscopists, people who are more looking into using a microscope rather than developing it. What super resolution technique is more suitable for me, and why should I use this and not that? And and they're trying to put some order into that with this paper. It's a very thorough paper. Recommend. It's a very good read, and a lot of what we will see a little bit later is going to be, excuse me, based on some of what was laid out in that paper. So yeah, here we see it again. Super resolution microscopy bypassed the diffraction limit of roughly 250 nanometers, which was previously thought to be impenetrable. I guess here is also worth as an aside, the resolution limit depends on the objective you're using. The resolution limit for a 10x objective is, for a simple 10x low NA objective, is going to be different than the resolution limit for a high NA oil immersion 63x 1.4 or what have you kind of objective, right? So these 250 nanometers are not the limit for a 10x objective. Those 250 nanometers are the resolution limit for the most high-end, high numerical aperture microscopes you can get, microscopy objectives that you can get. Okay, in that paper, there are a bunch of graphs. I'm not going to dive into that detail, but instead I extracted the main information here, and I placed this on the table where you have the main families of super resolution methods here organized from right to left, as the resolution scale, or the resolution improves from right to left. On the far right, we have the limit of what is possible, which would be represented by techniques such as confocal and deconvolution, which works on standard samples and is a proven and mature technique, but that requires a lot of averaging and long time exposures and things of that nature. And the first technique that surpasses this limit would be the first in terms of resolution, not historically, but would be the Eriscan super resolution technique, which is based on a confocal but collects light in a slightly different way, as we will see a little bit later. That has the advantages of a confocal microscope, which we will also see later, in its versatility, the ability to go deeper into a tissue, for example. Then the next technique that goes in the range between 120 and 60 nanometers would be lattice SIM. SIM stands for structured illumination microscopy. And yeah, here the main capability of that is the ability to go really fast with very low dosage of light and very little photo bleaching. The next technique that allows a resolution between 30 and 60 nanometers, typically these borders vary a little bit. STED in principle can also go down to a few nanometers. Is stimulated emission depletion microscopy, where the confocal, again, it's a point scanning techniques, is sharpened to a smaller probing volume, and that is raster scanned across the sample, as we will see also in an example. The most accurate, debatably, technique is the localization microscopy, where the position of individual molecules as they turn on and off is determined, and then the collection of these positions are then displayed as a map. And since it's possible to pinpoint the center of a molecule with more accuracy than to resolve it. If you're looking at an individual molecule, you'll be able to pinpoint it to its center with more accuracy than you can resolve the individual contours of that molecule. But if that molecule is decorating a structure and you pinpoint each and one of them individually, all you have to do then is connect the dots and get a super resolved image of the object that is stained with these fluorescent molecules. We will see that also in a little bit more detail, and perhaps the examples are going to be clearer to understand what is lying behind these techniques. So we're going to talk about three main classes of methods. One is the point scanning techniques, which encompass the Aries scan super resolution here to the right, and stimulated depletion here, the second one from the left. Then we're going to look at wide field based techniques, mainly lattice sim, the third column here. And then finally, we're going to look at localization based techniques, which are the ones on the far left. So if you look at point scanners, it might be interesting to reapproach, what is a point scanner and how does a confocal work to begin with, in order to understand how do we accomplish the super resolution with these systems. And the idea here is to use the confocal principal idea, some detectors, lasers, and fluorescence to construct a confocal microscope. As we can see here, this is one of those confocal microscopes from the 80s, all black and white with a cathode ray tube monitor. And the principle of it is as follows. We would illuminate the sample with a laser typically and collect its fluorescence. And the point there is that this illumination is not illuminating the entire sample, but it is focused through the objective onto the sample. The light that is emitted in that hourglass type shape is collected by the same objective, then separated from the excitation light and reaches a photomultiplier tube, the detector as seen on the top here. What we can also see in this picture is that light that is coming not from the focal spot, the blue and the green dotted lines here, they also reach that detector, and that is unwanted light. And if there is any way we could suppress that light, and that is accomplished physically by placing a little hole, a pinhole, in the conjugate focal plane, hence the name confocal, and only the light that is focused there will completely pass that pinhole and reach the photomultiplier tube. So since only that focal spot is imaged, it has to be scanned, raster scanned across the specimen to produce a full optical image. So what you can do is you can take that object and scan it around, or you can place a scanning mirror, which is what is done today, in front of the objective and scan that spot across the sample in order to obtain an image. The resolution of that optical section or of that microscope is controlled by the numerical aperture of the objective lens, the wavelength, and the pinhole diameter. If we take a look how the image is generated, here in comparison, we have the laser wide field, which generates an image in an instant shot. We collect everything at the same time, albeit with all the haze and the blur that is around that sample. On a confocal, as we're scanning and suppressing light that is coming from the different planes, the acquisition looks more or less like this. You see it's a little bit slower, but the image has a lot more contrast. Because of that, the confocal imaging is also intrinsically 3D, and that is the main use for a confocal microscope. If you look at these images here, those are 3D volumes collected from a brain or from a piece of a flower, I think that was. Yeah, so in order to generate 3D images. And if we take a look here again at the definition of, the resolution in a confocal is dictated by the pinhole diameter. So the first question is, hey, if I make this pinhole smaller and smaller, will I be able to obtain more and more resolution? And the answer is yes. So strictly speaking, any confocal microscope is already a super resolution microscope. The point, however, is that the price to pay for closing that pinhole is an extreme loss in intensity. So you would sacrifice 95% of your light in order to get a 40% increase in resolution. And that, for the vast majority of confocal studies, is prohibitively large, that loss. So you would have to fry your sample with laser and bleach the hell out of it before you're able to collect a super resolved image on a confocal instrument. So around that time, and we're talking about the 80s, there were already ideas in place to think about, well, how can we at the same time close the pinhole and collect all the light? And then recently it was possible to design a detector that is not a single pinhole and a detector, but is an array of detector pinholes that are placed exactly in the confocal plane. So when we're using this array of detection, each camera, each little element of that camera, each pixel on that camera acts like a pinhole, like a closed pinhole. The advantage there is that each one of these cameras is collecting a closed pinhole image, albeit from different angles, that is exploring all that enhanced resolution. But since I'm placing these cameras all over the place, I'm not rejecting any light in the end. So I'm collecting all the light, but generating many images from different angles, all with a closed pinhole. The exercise I need to do now is to find a way to computationally convert the individual images into one single image that benefits from that super resolution. And this is what area scanning does. And on an instrument, it looks like you see here on the screen. You would have that special detector array that's composed of many hexagons, concentrically organized, that would image the confocal plane and collectively generate a high resolution image. Let's take a look at one of these high resolution images and what that means for research. And we have here a telomere replication with and without, in this case, without a telomere maintenance protein RTL1. And what these researchers were interested in is, like, in the absence of this protein, how do the telomeres of these chromosomes look like? Are there bifurcations, for example? And if we juxtapose the two images, to the left, the confocal image, and to the right, the area scanning image, we immediately appreciate, the little dots that were not possible to see the difference between, the telomere bifurcations now become visible. And if you're looking into these minute differences, a super resolution technique is going to be enabling for you to be able to resolve these things. So they were just waiting for any technique that gives you a little better resolution than confocal and better signal to noise ratio, so that you can distinguish between these two objects. Okay, then are there other ways to increase the resolution of a confocal instrument? And the answer there is yes. You can engineer on the detection side, which is what we've seen in the previous slides by looking at the area scanning, but you can also engineer on the excitation side. And that is the Nobel Prize winning technique from 2014, or one of the techniques from 2014 that received the Nobel Prize, which is stimulated emission depletion, STED, which works as follows. So instead of focusing, or in addition to focusing the light to the tiniest possible spot in the sample, there is an additional beam, that's the depletion laser that you see here, that is also focused into the sample, but it goes through a faceplate, a beam shaper, if you will, that will not form a ball of light, but a donut of light. And that donut of light has that, you know, little hole in the middle where the blue light would completely pass through, and a circle of light that would inhibit fluorescence, or it would actually do the simulated depletion. It's fluorescence, but sort of instant. That would inhibit that fluorescence to be collected with a detector. So by increasing the intensity of that donut, I can make the hole of the donut smaller and smaller, like sharpening a pencil, and in that way scan over the sample with a smaller focus, then collect all the light with a pinhole onto the photon detection system, and in that way generating a super resolution image, typically down to 30 nanometers. We see here in the Charmele paper, there is a diagram showing, you know, a decision diagram, and one of the aspects of it is for example, low cost 2D STED, as seen here, can be added to many microscope stands. And what you see here on the left is a typical image of I think spectrin in this case, where an image was generated without the depletion beam here seen on the top. This is the confocal image, and then with the depletion beam, see here on the bottom, where you can resolve these structures that are sort of dozens of nanometers in separation from one another. Then the next family of techniques would be the wide field based techniques using structured illumination microscopy and lattice structured illumination microscopy. Very recent developments in that field. And the way it works from a physicist's perspective is very easy. What we're doing here is we're illuminating the sample with a pattern, with a grid, or with a lattice, and in that way generating beat frequencies. Beat frequencies, as you can see here in the image, are these moire patterns, the bigger undulated objects that you see, where the the blue and the black grids overlap. And these beat frequencies are down conversions of higher frequencies. In physics, this is called heterodyning. What you're doing is you're converting frequencies that you cannot observe because they're too high frequency or too small in space. You are modulating them with a known frequency, a frequency that you can resolve, and then looking at the beat frequencies. And all you have to do then is find a way to untangle these results and back calculate what the original image looked like. That is a little bit cumbersome detail to go through here in detail, but in a hand waving way, this is what happens. You're taking 3 images. You're taking the image with a pattern, and you're moving that pattern around. Well, you're taking several images of that pattern moving around, and then you're decomposing it in Fourier space and frequency domain in order to reconstruct the image that was generating these moire fringes. In practice, it looks like this. The top row here shows a zoomed in view of the lattice of that patterned illumination that is being scanned across a mesh of tubulin, and here typically you would record 10 images or so, which can take somewhere around 50 milliseconds or so to collect these images. And then the reconstructed images on the bottom. Then on the left side, we see the super resolution reconstruction, and on the right side, how it would look like under a wide field illumination. And yeah, the advantages of this technique is that it's extremely fast, that you can image at 255 frames per second. And you can see how that looks like here, again, by looking at the mitochondria moving around a meshwork of endoplasmatic reticulum. And I think for me as a microscopist, the most important thing I see here is the capability of recording like 20 seconds worth of images and thousands of frames without any noticeable photo bleaching. There is some photo bleaching, but it's not a lot of photo bleaching. And that is going to produce a wealth of data for me to look at the dynamics of mitochondria or compartments of the endoplasmatic reticulum and the diffusion, interdiffusion between these two organelles. We can drive lattice sim, or sim squared as it's called, to yield resolutions down to 60 nanometers, as seen here in relatively technical samples where we have a DNA origami that was specifically designed to have a specific separation between the two dyes that are in the 5' and the 3' end of that construct. And you have there to the left 80 nanometer separation, and to the right you have 60 nanometer separation between the two dots, and they're well resolvable in all kinds of angles with lattice sim squared. Of course, this also works in living organisms. Here again at full speed, looking at tubulin and endoplasmatic reticulum and the co-diffusion. And if we take a look at the zoom in here where this square is, and we try to figure out, what's the smallest distance that we can still see between individual filaments, we can see here we are achieving easily a scale that is below 100 nanometers with this technology. As another example, we see here the synaptonemal complex stained with 3 different dyes. One that would side the chromosomes, and the other ones that would be in the center of this Velcro-like structure between the two. And yeah, here seeing that you can easily distinguish in multiple colors the different layers that are within that structure. They are separated by, here in this case, 50 or so nanometers between the individual spots. So this entire structure under a normal confocal microscope would appear as one blur, and it would be virtually impossible to distinguish between the individual layers that are underneath that structure. Okay, then we're approaching the end of our time, and I have to rush us through the localization-based techniques. The idea here is that we're going to image tubules again in this example, but we're not going to look at all the dyes simultaneously. What we're going to do here is we're going to somehow provoke the molecules to turn on and off, either chemically or using photonics. And then instead of detecting all the molecules on at the same time, we will turn on each lamp individually, as shown here on the top right. You can see the positions of individual molecules, and then reconstruct the object, not from the big flash of light, but the pinpointing of the center of these molecules, and then plotting all those together. And what you see then is on the left side, this junction of two tubules here, and you can see easily the separation between two of these tubules with spaces between them of 20 or so nanometers. Another example is shown here. What you see here is a nuclear pore complex. So this is the whole orange thing is a nucleus, and these granules you see there are the individual nuclear pore complexes. Everything that enters or exits the nucleus has to go through that little door, or all the big molecules that go in and out, all the information, RNA, etc., that go in and out of the nucleus will have to at some point interact or pass with a nuclear pore complex. So it's a very important part of the biology is happening there. And if we turn on the the single molecule localization microscopy technique, we will be able to resolve not only in space how these nuclear pore complexes look like, but we will have access also to the possibility of counting how many molecules are in there. Let me show you an example. So first, we get rid of the resolution limit, and we're able to look at individual nuclear pore complexes. And second, we're able to go into specific nuclear pore complexes and count how many molecules are in there. And we will see here that each nuclear pore complex for this specific protein is composed of 4 subunits of that, separated by a larger cleft. We have one molecule here, another molecule, then a cleft, one molecule, molecule, cleft, molecule, molecule, cleft, and so on. And there are 8 of them. So you're able to see how individual molecules are organized around a nuclear pore complex, not crystallized and analyzed under an electron microscope or an X-ray diffractometer or something like that, but in the actual intact cell, or stained cell, or intact cell. At least the morphology of the cell is somewhat intact. This can be done also in multiple colors. Seen here, beta and alpha tubulin stained with different fluorophores and detected with 2 cameras that are aligned. And we see here maybe an important notion of super resolution microscopy or molecular resolution microscopy is that there is no colocalization on the molecular level. Where there is one molecule, there can't be another molecule. They cannot physically overlap because they have a certain size. And since this super resolution method affords us to observe objects on that scale of length, we are able to see that, you know, whereas one molecule, there can't be the other molecule unless it's on top of it, but there can't be the other molecule. It will have to be somewhere lying on the side of it. And this is nicely exemplified here. You will see very rare cases only where 2 tubules overlap or something like that, where you would have, you know, yellow pixels, but the alpha and the beta tubulin are nicely separated here. Yeah, this is also possible in 3D, single molecule localization microscopy by modulating the PSF. I won't bother you with those details here, but as an example, we have 2 centrioles imaged here in 3D, and they can be, and I like this example because you can look through one centriole and see the other one. All done with molecular precision here. Okay, we're approaching the end of the presentation. I would like to finish off with a summary and some examples for literature. The number of publications that explicitly mention super resolution is in the thousands per year. No, well, more than 1,000 per year. And yeah, it's a thriving field. I think that the benefits from super resolution microscopy are quite obvious. The more you see, the more detail you observe, the richer your research can be, the more knowledge you will obtain from the biology, biophysics, and what have you, and you'll be able to describe the systems better and find better diagnosis, better ways to interact with the molecules of interest there. Microscopy techniques, super resolution microscopy techniques are one technique among a broad range of tools. So microscopes are, not all microscopes are super resolution microscopes. There are microscopes that also offer, you know, a zoomed out view of animals and of organisms, where you don't want to look at the individual detail, but you want to look at the entire specimen. And on the other side of microscopy, you have also all the electron microscopes and all the sub-nanometer down to sub-nanometer resolution microscopes. There is a broad palette of microscopy that can be employed to answer biologically relevant questions. One of them is super resolution microscopy. Finally, I'd like to finish off with providing some interesting links. One of them is the link to the Harvard Center for Biological Imaging and our YouTube page, where individual tutorials can be visited, and also the ZEISS campus site, which has a number of interactive tutorials, for example, on the topic of super resolution microscopy, but also many other topics. And with that, I would like to thank you, and yeah, hope for some interesting questions. Thank you very much.
Excellent, Christian. Thank you so much for a really good overview of the suite of super resolution microscopy techniques out there. I'm now going to invite questions